Okay, so this next chapter deals with protein metabolism, which really just means, or the part that we're going to focus on, is translation, meaning how proteins get made in the cell. But before we talk about that, let's talk about the genetic code. So the genetic code is what it sounds like. It's the code that the cells use to take a given nucleic acid sequence and turn it into an amino acid sequence. And this is, in many ways, a bigger problem to crack than the problem of transcription or DNA replication, because making a copy of DNA or making an RNA copy of a DNA sequence is relatively easy because there are only four bases. They base pair in a very specific and predictable way. So the mechanisms for those processes involve a lot of proteins and a lot of different components, but the overall process is relatively simple. Whereas with translation, you have four nucleic acids and 20 amino acids. So the question is, how do you encode those 20 amino acids using just the four bases of DNA and RNA? So first of all, just a little bit of background to talk about how this was figured out, because it was involved a lot of different people over many years working in parallel to figure it out. But some of the earliest discoveries were, first of all, where protein synthesis even occurs, because of course it happens in the cell, but the question is where in the cell. And so the fact that proteins are synthesized by ribosomes was an early and important discovery, and it was basically figured out by using radioactively labeled amino acids and then looking in the cell or fractionating a cell and figuring out which part of the cell contained those newly formed proteins that had the radioactive amino acids in them. And so it was eventually figured out that it was the ribosomes that were the components that actually assembled the proteins. Some of them are attached to the endoplasmic reticulum, some of them are free ribosomes, and of course that's just in eukaryotes. In prokaryotes, they also have ribosomes, but their ribosomes are just free. But those are the parts that actually make the proteins. And then the idea that you can't just use any old amino acids to make a protein, you have to use activated amino acids. And an activated amino acid is one that is attached to a special kind of RNA molecule, which we'll be talking about more later, called a transfer RNA or tRNA. And there are enzymes called amino-acyl tRNA synthetases, and their job is to take an amino acid and a specific transfer RNA that's specific for that amino acid, and using ATP for energy, combining the two together. And that's this amino-acyl tRNA is the activated form of the amino acid that you can use to make a protein with. And then there was the problem of how do you actually get the amino acid and the mRNA sequence to correspond. And so it was obvious early in this research that there must be some sort of adapter, and that told the ribosome which amino acid to add, and it became clear pretty fast that it's the transfer RNA itself that is the actual adapter molecule. So we'll be talking about how that works. Now, the actual genetic code, though, in other words, which bases and which combinations code for which amino acids, took several years to figure out, and again involved a lot of different groups. So some pretty fundamental questions come up when you start looking at the nucleic acid bases and the 20 amino acids. Because there's only four bases and 20 amino acids, you can't have a simple one-to-one code, because then you could only have four amino acids. So if there were codons or combinations of bases that only involved one base, then you could only have four amino acids in a protein. So you can make a more combinations of bases by increasing the number of so-called letters in a codon. If you had two bases per codon or two letters per codon, you could have up to 16 combinations because there are four possible bases at each position. So the total number of combinations is 4 times 4. But that only produces 16, and that's not enough because we know that proteins are made of a combination of 20 different amino acids. So a 3-letter combination is the only one that gives you, or the smallest number that gives you enough possible codons. So with 3 bases per codon, you can make 64 different codons because at each position you can have any of the 4 different bases, and so there's 4 times 4 times 4 possible codons. So we now know that the genetic code includes those 64 combinations. So that's more than enough to encode all 20 amino acids. And that is in fact exactly what the codon or the genetic code looks like. You've probably seen this table before. This is the codon table, which shows all 64 codons that you can make using the 4 bases of an RNA molecule and the 20 amino acids that you can get from those. And so the way you read the codon table, the first letter of a codon is read on the left-hand side here on this column. The second letter is read across the top, and then the third letter is one of the 4 bases within each little box. So for example, if you wanted to know what the codon GAC coded for, you would start here. You would say, okay, the bottom row corresponds to G at the first position. The third column corresponds to A at the second position, and so it's going to be somewhere in this box. And so here's GAC, and it corresponds to the amino acid aspartic acid or aspartate. So all 64 codons can be looked up this way, and you can see that all of them, except for 3, have an amino acid associated with them, and so we'll talk about those 2 in a minute. But again, how was this code actually figured out? A bunch of different people were working on this, and it involved a lot of different interesting and kind of clever experiments. The story of DNA structure often gets a lot of attention because it was a very dramatic story involving just a small number of people working over a short period of time. But the genetic code story, in some ways, even though it lasted longer and involved a lot more work, in some ways is a lot more interesting. So, for example, this guy Marshall Nuremberg and his team actually worked out the very first codon. And the way they did it is they made these artificial RNA molecules that just consisted of one base. So the first one they tried was a polyuracil. It was just a bunch of U's attached 5' to 3', just like a regular RNA, but with just uracil, no other base in there. And they mixed that with an E. coli extract. So these are E. coli bacteria that they just lysed, in other words, ground them up into their components, mixed them together with this polyuracil, and then put that mixture into 20 different tubes, each of which contain a different amino acid. And then they looked to see what they got. And in this case, all they got was a protein in the tube that contained the amino acid phenylalanine. So out of all these other tubes, no new proteins were formed. But in this one that contained phenylalanine, they got a protein that just consisted of a bunch of phenylalanine amino acids linked together. So the explanation for that is that this codon UUU must be the codon for the amino acid phenylalanine. And they did that for CCC, which turned out to be the codon for proline, and AAA, which turned out to be the codon for lysine. For technical reasons, you can't make a mRNA out of polyguanosine because polyguanosine makes these tetrahelical structures that can't be translated. But those first 3 codons were worked out using this experiment. And then later, this guy Gobind Khorana and his team worked out some more codons by making slightly more complicated RNA molecules. This time, instead of just one base repeating, he made combinations of 2 or 3 or 4 bases. So for example, this mRNA sequence is made by just taking a repeating 2 nucleic acid sequence of adenine and cytosine. So you have just AC, 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 AC. Again, you mix that with an E. coli extract. Presumably the E. coli extract has ribosomes, has transfer RNAs, has everything else that you need to make a protein other than mRNA. And then mix that with the 2, this time containing more than just one amino acid, so you can do this with all 20 amino acids. And in this case, when they did this, they got a protein which consisted of just 2 amino acids, threonine and histidine. And because the threonine was the first one in that sequence, the codon for threonine must have been ACA, because that's the first 3 bases of this mRNA, whereas CAC, that's the second 3 bases of this mRNA sequence, must code for the amino acid histidine. And then just by doing this with other combinations of bases, they and other labs worked out the actual code. So for every amino acid, a 3 base code was determined. And this code turns out to be almost universal. So if you look at the amino acids, the codon table for almost any species, it's pretty much the same. There are a handful of exceptions. Mitochondria, for example, they have their own genes that they express. They make some of their own proteins, and there's a couple of places where the code does not quite match up to the nuclear genome. But when you look at eukaryotes, prokaryotes, pretty much every organism that has been looked at so far, this codon table is universal. And this is important for a couple of reasons. One, it's pretty strong evidence that all living things are descended from a common ancestor. And two, it's really important for biotechnology, because what this means is you can take the nucleic acid sequence of, let's say, a human gene and put it into the genome of, let's say, a bacteria or a yeast cell, and that cell will make the exact same protein that the human cell would have made, because the yeast cell and the bacterial cell and the human cell all have the same genetic code. So this code is pretty fundamental to life as we know it. But anyway, we know that it does have a 3 base codon system. Again, that's necessary because of the number of amino acids. So each codon corresponds to, or contains 3 bases. Each codon corresponds to 1 amino acid, although it doesn't work the other way around. So you can see here that each amino acid can have more than 1 codon. We'll talk about that later. But there is, every codon has only 1 amino acid that it can code for. The genetic code is read in the 5' to 3' direction, so that refers to the orientation of the mRNA. So the mRNA is synthesized, remember, in the 5' to 3' direction. So the 5' end of an mRNA is the first part of the mRNA to get made, and the 3' end is made last. And the synthesis of proteins starts at the 5' end and goes to the 3' end. We'll talk about that later, but this is the direction that the code is read. So the anticodon, which is the part of the transfer RNA that recognizes the codon, binds to the codon, and so the first codon will be at the 5' end, the last codon will be closer to the 3' end. Now the anticodon of the transfer RNA, because it's complementary to the codon of the mRNA, is antiparallel, and so we read the codon, in this case this codon is AUC, but the anticodon we would say is GAU, because traditionally the 5' end of any nucleic acid sequence is first, but that's so sometimes you'll see anticodons written sort of backwards like this. Another important feature of the genetic code is that it is non-overlapping. So each 3-letter codon is its own codon, and the second codon in a sequence starts with the fourth base, and the third codon starts with the seventh base, and so on. And as opposed to an overlapping code, so this would be what an overlapping code might look like, where the first codon here would be AUA, and the second codon would start with the second base, where the second codon would then be UAC, and the third codon would be ACG, and so on. It perhaps could have been built that way, but that's not the way the genetic code works. It is a non-overlapping code. Also, wherever translation starts, and translation doesn't start necessarily at the 5' end of the mRNA, so it starts somewhere internal to the mRNA. But wherever the first codon is, and we'll talk about initiation of translation later, but wherever that first codon begins, that establishes what's called the reading frame. And the reading frame then continues for the rest of the mRNA. So any given nucleic acid sequence can have at least 3 reading frames. And so here, this first reading frame starts here with this U, and so it goes UUC is the first codon, UCG is the second codon, GAC, and so on. Whereas down here, if the first codon was one base moved toward the 5' end, or 3' end, then this would be its reading frame. So UCU would be a codon, CGG would be a codon, and so on. And then this would be the third reading frame, where now you've got these two U's as the end of the previous codon, and then CUC, GGA, CCU, and so on. So any given sequence has 3 reading frames. If you move down one more position, you're back to the first reading frame again, where this is the first base of the first codon, and so on. What that means is any mutation that causes either an insertion of a base or a deletion of a base are going to create what we call frameshift mutations. So a frameshift mutation, like here, if let's say you have an insertion into this position here. So right here, between this U and this C, there's a mutation in which there's a mistake during DNA replication, let's say, and a C gets stuck here between that U and that A. Well, now that codon that used to be UAC is now UCA, but not only is that codon wrong, but all the ones downstream from it, all the ones in the 3' direction. So this codon that used to be GGA is now gone. It's now out of frame. And the new codon is CGG and so on, and that's going to create a completely different protein. All these codons now code for completely different amino acids. Deletion would do the same thing. A deletion mutation would shift the reading frame to the left, but what that also means, and the way that this was sort of proven that the code was non-overlapping and then each codon has 3 bases, is by looking at combinations of insertion and deletion. So if you have an insertion and a deletion in the same gene, if they're reasonably close together, they kind of cancel each other out. So here we've had an insertion at this position, so between this A and this G, and then a deletion here. So between this U here basically got deleted. And so these two codons are now wrong. So this codon used to be GCC, now it's AGC. This one used to be UAC, now it's CAC. But it's only those two codons that are mutated. The rest of them downstream will be back in their original reading frame. Also, if you have any number of insertions or deletions that occur in multiples of 3, you can also restore or rescue a reading frame or frameshift mutation. So 3 insertions in a row or 3 insertions anywhere will shift the reading frame back to its original reading frame for at least part of the protein. Now, like I said, all 20 amino acids are represented in the codon table, but there are 64 codons, which means there's more than you need. And the only ones that are really exceptional in this way are So there are some special codons within the codon table. There is one initiation codon. So the codon AUG in every organism codes for the amino acid methionine, and it also is the first codon of every protein. Now later on we'll see that most proteins the methionine gets chopped off, but that's always the first codon to get added, so it's called the initiation codon. And there are 3 codons that do not have any amino acid associated with them. They are called the stop codons. So when translation or when a ribosome gets to one of these 3 codons, UAA, UGA, or UAG, it stops. It stops translation, and that's the end of the protein. Also, any mutations, of course, then that introduce stop codons are especially problematic, because it means that if a mutation creates a stop codon in the middle of a protein or toward the beginning, then any amino acids that are supposed to be present downstream from it get eliminated. One other important feature of the genetic code is that it is degenerate. Now, that does not mean that it is flawed or ambiguous, because, again, every codon represents one and only one amino acid. So if I give you a codon, you should be able to find the amino acid that it goes to. But it does not go the other way around. That's what degenerate means. For each amino acid, for some amino acids, there can be more than one codon. In fact, for most amino acids, there is more than one codon. Methionine and tryptophan are the only 2 that only have one codon associated with them. The rest have at least 2. And then when you get to arginine, leucine, serine, they have all the way up to 6. Now the reason that some amino acids have more than one codon is because most of them have more than one transfer RNA. So most amino acids have multiple transfer RNAs that they can be bound to, or that they can be activated by. But another reason for degeneracy of the genetic code is that this last position of the codon, the third base of the codon, is sometimes called the Wobble position, because the position of the anticodon, that the base on the anticodon that binds to the Wobble position, can be a little ambiguous. So different bases have more or less Wobble properties than others. But for example, if you have a codon like this, CGA, an anticodon that's GCI. I, by the way, is a modified nucleic acid or nucleotide that is only found, or is mostly found, in transfer RNAs, and it's has the property of being able to base pair more non-specifically than the normal bases. So the codon GCI, or actually ICG, if we're reading from the 5' end, can bind to at least these 3 codons, so CGA, CGU, CGC, because it's in the Wobble position and because it's a little bit less picky about what it binds to than other bases. So these are other bases that can wobble. So if you're actually there's 2 that don't. So if your 5' position of the anticodon is a C or an A, there will be no wobble. So C in that 5' position only base pairs with G. A in that position only base pairs with U. U at the 5' end of the anticodon can hydrogen bond with either A or G, so there's some wobble there. A G in the 5' position of the anticodon can hydrogen bond with C or U, so it has some wobble as well. And again, inosinate is the modified version, purine, that is found in tRNAs, and it's the least picky of all, so it will bind to A, U, or C. So that's the reason why the code is degenerate. So next time we will talk about the details of translation itself.